Hello there, and welcome to the Sleepy Science Channel. Tonight we'll be exploring the quiet, hidden world of carnivorous plants. These remarkable organisms dwell in forgotten corners of the earth, in bogs and wetlands, on mountain tops and tropical forests. They are patient hunters, living sculptures that have turned the very notion of what a plant can be on its head. While most plants reach toward the sun for energy alone, these extraordinary species have learned to draw nutrition from another source entirely: the insects and creatures that wander into their grasp. The world of carnivorous plants is one of elegance and adaptation, of beauty-concealing function, and of patience measured not in moments, but in days and weeks. If you enjoy these quiet explorations, I invite you to like and subscribe, or share a simple thought down in the comments. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is rest, settle in, and allow the gentle fascination of these botanical wonders to carry you softly into sleep. Let's begin. The origins of carnivory. Carnivorous plants evolved independently across the globe in nutrient-poor environments. In the grand story of life on Earth, plants typically follow a simple formula. Roots draw water and minerals from soil, leaves capture sunlight for energy, and together these processes sustain growth. But in certain places, the soil offers almost nothing. Bogs and marshes, sandy heaths and rocky mountainsides, tropical plateaus where rain washes away nutrients before they can accumulate. In these harsh landscapes, some plants found another way. Over millions of years, in at least 5 different plant families scattered across the world, evolution crafted the same remarkable solution: carnivory. Leaves transformed into traps. Ordinary plant structures became tools of capture. Digestive enzymes, similar to those in animal stomachs, began secreting from modified leaf surfaces. This convergent evolution, where unrelated species independently develop similar traits, speaks to the power of adaptation. When the environment demands innovation, life responds. These plants did not abandon photosynthesis. They still capture sunlight, still produce their own sugars through the ancient chemistry of chlorophyll. But they supplement their diet with nitrogen, phosphorus, and other vital nutrients extracted from the bodies of insects. In doing so, they transformed from passive recipients of whatever the soil could offer into active participants in the cycle of life and death. They became hunters that never move, predators that never chase, killers cloaked in beauty and stillness. The evolution of carnivory required multiple coordinated changes. A plant cannot simply develop a trap without also developing the means to digest what it catches and absorb the resulting nutrients. Each step in this evolutionary journey had to provide some benefit, no matter how small. Perhaps it began with sticky leaves that accidentally trapped insects, which then decomposed on the leaf surface, releasing nutrients that the leaf happened to absorb. Over countless generations, natural selection favored plants that were slightly better at trapping, slightly better at digesting, slightly better at absorbing. The process was gradual, each improvement building on the last, until finally, a fully functional carnivorous plant emerged. Venus flytraps closed their jaws in a fraction of a second. The Venus flytrap is perhaps the most iconic of all carnivorous plants, and for good reason. Native to a small region of coastal wetlands in North and South Carolina, this species has captivated human imagination for centuries. Its leaves form hinged traps, each one resembling an open mouth lined with interlocking teeth. But these are not jaws of bone or keratin. They are living leaf tissue, flexible and responsive, capable of one of the fastest movements in the entire plant kingdom. When an unsuspecting insect lands on the trap's surface, it brushes against tiny trigger hairs. Each trap contains several of these hairs, and the plant has evolved a remarkable counting mechanism. A single touch means nothing. It could be a raindrop, a falling twig, a false alarm. But when two hairs are touched within about 20 seconds, or the same hair is touched twice, the trap recognizes prey. In that instant, something extraordinary happens. The trap snaps shut in less than 1/10 of a second. The movement is so swift that slow-motion cameras are needed to capture its full sequence. The mechanism involves a rapid change in the curvature of the leaf, powered by the sudden movement of water and changes in cell pressure. It is not a muscle contraction, for plants have no muscles. It is hydraulics and tension, geometry and fluid dynamics, all orchestrated by a plant with no brain, no nervous system, no consciousness as we understand it. Yet it counts, it remembers, and it acts. Once closed, the trap does not immediately seal completely. The interlocking teeth form a cage, allowing tiny insects to escape while larger prey remains trapped. This is intentional. Small insects offer little nutrition and are not worth the energy cost of digestion. But if the prey is substantial and continues to struggle, the trap senses this movement through continued stimulation of internal trigger hairs. Only then does it seal tightly, forming a stomach in which digestive enzymes flood the space, breaking down the insect's body over the course of 5 to 12 days. The physics of the Venus flytrap's closure has fascinated scientists for decades. Recent research using high-speed cameras and mathematical modeling has revealed that the trap operates through a snap buckling instability, similar to how a contact lens can flip inside out. The leaf stores elastic energy by maintaining a curved shape under tension. When triggered, the leaf rapidly flips to a different stable configuration, releasing that stored energy. This mechanism allows for incredible speed without requiring the plant to generate force quickly, which would be impossible with plant cells alone. The traps can only close a limited number of times. The Venus flytrap's dramatic snap comes at a cost. Each trap can close only a handful of times before it dies. Some estimates suggest 3 to 5 closures for feeding, though the trap may snap shut more times if triggered falsely without prey inside. Every closure uses energy. The rapid movement damages cells. The production of digestive enzymes is metabolically expensive. After each feeding, the trap must recover, and with each cycle, it grows weaker. This limitation makes the Venus flytrap's counting mechanism even more remarkable. The plant cannot afford to waste closures on false alarms. Rain, wind, falling debris, these must be filtered out. By requiring 2 touches in quick succession, the plant ensures that only moving, living prey triggers the trap. It is a simple algorithm, but devastatingly effective. In the harsh environment where nutrients are scarce, this efficiency means the difference between survival and starvation. When a trap has exhausted its usefulness, the plant simply grows new ones. Throughout the growing season, fresh traps emerge from the center of the rosette, young and eager, while older traps around the edges blacken and wither. The plant continually renews itself, replacing spent traps with fresh ones, maintaining its capacity to feed. This cycle of growth and decay, of new beginnings emerging from exhaustion, mirrors the rhythms of life itself. To watch a Venus flytrap in the wild is to witness patience punctuated by violence. For days, the traps sit open, motionless, waiting. Then, in an instant, the snap, the struggle, the slow sealing. And finally, stillness again, as digestion proceeds in silence, hidden within the closed leaves. It is a reminder that even in the quietest corners of nature, drama unfolds. invisible to those who do not take the time to watch. The energy economics of the Venus flytrap are fascinating to consider. Each trap requires significant resources to build and maintain. The trigger hairs must be exquisitely sensitive. The hydraulic system must be primed and ready. The digestive glands must be prepared to secrete enzymes on demand. All of this happens in a plant growing in one of the most nutrient-poor soils imaginable. The Venus flytrap is making a calculated gamble with each trap it produces, betting that the nutrients gained from captured prey will exceed the cost of building and operating the trap. In the acidic, waterlogged soils of coastal Carolina bogs, this gamble pays off. Pitcher plants create death traps filled with digestive fluid. While the Venus flytrap actively grabs its prey, pitcher plants take a different approach. The pitfall trap. These plants grow modified leaves that form deep, vase-like structures, often beautifully colored and patterned. The opening, called the peristem, is often slick with waxy secretions or downward-pointing hairs that make escape impossible. Inside, a pool of liquid waits. Part rainwater, part digestive enzyme, part decomposition fluid, teeming with bacteria that aid in breaking down prey. Insects are lured to the pitcher by nectar secreted around the rim, by bright colors that promise food or shelter, by scents that mimic fermenting fruit or carrion. They land on the peristome, perhaps sip the nectar, and then lose their footing. Some pitchers have surfaces so slippery that insects slide immediately into the depths. Others have downward-pointing hairs that allow entry but prevent any upward climb. Once inside, the struggle begins and almost always ends in drowning. The fluid within a pitcher plant is a complex ecosystem in miniature. Different species of pitcher plants host different communities of organisms. Some contain only digestive enzymes produced by the plant itself. Others host bacteria that help break down the prey, creating a symbiotic relationship where both plant and microbe benefit. Still others are home to specialized insects that have evolved to live within the deadly fluid, immune to its effects, feeding on the drowned remains of less fortunate visitors. Pitcher plants can be found across the world, from the tropical mountains of Southeast Asia to the temperate bogs of North America. Each region has evolved its own pitcher plant lineages, independent solutions to the same problem of nutrient scarcity. The Asian pitcher plants, genus Nepenthes, produce elaborate hanging pitchers that can grow larger than a football, some capable of capturing not just insects, but frogs, lizards, and even small mammals. The North American pitcher plants, genus Sarracenia, grow as vertical tubes from the ground, some standing waist-high in autumn bogs, their colorful hoods arching over openings that promise shelter but deliver death. The architecture of a pitcher plant is a masterpiece of evolutionary engineering. Consider the peristem, the rim of the pitcher. In many species, it is ribbed with grooves that channel water away during rain, preventing the pitcher from overflowing and losing its digestive fluid. The surface is often coated with microscopic wax crystals that create an impossibly slippery surface. An insect's claws, designed to grip nearly any surface in nature, find no purchase on these crystals. The waxy coating also repels water, ensuring that rain beads up and rolls off, rather than dissolving the trap's deadly slickness. Below the peristem, many pitchers have a zone of downward-pointing hairs. These hairs are rigid and sharp, angled like the teeth of a funnel. An insect can easily push past them on the way down, but any attempt to climb back up drives the hairs into the insect's body, impaling it, or at least blocking its progress. It is a one-way valve, elegant in its simplicity and brutal in its effectiveness. Some pitcher plants are large enough to trap rodents. The largest pitcher plants grow in the jungles and mountains of Borneo, Sumatra, and the Philippines. Nepenthes raja, often called the king pitcher plant, produces pitchers that can hold more than 3 liters of fluid. These botanical giants grow on exposed ridges and summits, where nutrient-poor soils make conventional nutrition nearly impossible. Their size is not merely for show. It reflects their ability to capture substantial prey. Smaller pitcher plants subsist on ants, flies, and the occasional beetle. But Nepenthes raja has been documented capturing rats, shrews, and even small birds. Carcasses of vertebrates have been found within their pitchers, slowly dissolving in the acidic fluid. The plant does not actively hunt these creatures. It simply provides an enticing target, a scent of nectar and decay, a promise of food or water in a harsh environment where both are precious. When an animal leans over the rim to drink or investigate, the slippery surface betrays it, and gravity does the rest. The digestion of a large animal takes weeks. Unlike the rapid processing of an insect, which might be reduced to an empty exoskeleton in days, a mammal provides protein and nutrients that the plant absorbs slowly. The fluid within becomes a soup of decomposition, aided by bacteria and perhaps by specialized larvae that live within the pitcher without being digested themselves. These larvae break down the carcass mechanically, speeding the process and making nutrients available to the plant more quickly. To encounter one of these giant pitchers in the wild is to feel a strange reversal of expectations. We think of plants as passive, gentle, part of the backdrop of nature. But here is a plant that could theoretically capture a small pet, a plant with a mouth large enough to swallow your fist, a plant that smells faintly of death and sweetness. It is beautiful and unsettling in equal measure, a reminder that nature's solutions to survival can take forms we scarcely imagine. The discovery of Nepenthes raja in the mid-1800s caused a sensation in botanical circles. Explorers returning from Borneo brought back specimens and tales of enormous pitchers hanging from vines on remote mountaintops. Some accounts were embellished, speaking of pitchers that could swallow a human child, but the reality was extraordinary enough without exaggeration. Here was a plant that challenged the very definition of what plants could do, what they could be. Even today, new species of large tropical pitcher plants are occasionally discovered. The rainforests of Southeast Asia remain imperfectly explored, with remote regions still hiding botanical surprises. In 2007, a new species, Nepenthes attenboroughi, was discovered on a remote mountain in the Philippines. Its pitchers rival those of N. raja in size, and its discovery reminded the world that even in the 21st century, nature retains mysteries and new wonders wait to be found in the least accessible corners of the earth. Sundews glitter like jewels with sticky traps. If pitcher plants are the pitfalls of the carnivorous world, sundews are the flypaper. These plants produce leaves covered in tentacles, each one tipped with a glistening drop of mucilage. In the morning light or when backlit by the sun, a sundew sparkles as though covered in dew, hence the name. But this is no morning moisture. It is a deadly glue that ensnares any small insect unlucky enough to land upon it. The tentacles are living structures, connected to the leaf through vascular tissue. When an insect touches the mucilage, it sticks immediately. The struggle begins, and as the insect thrashes, it contacts more and more tentacles, becoming ever more entangled. Some sundews have tentacles that can move, slowly bending toward the prey over the course of minutes or hours, wrapping around it like fingers closing into a fist. The entire leaf may even curl inward, bringing more digestive surface into contact with the captured meal. Sundews are found on every continent except Antarctica, a testament to their success. Some are tiny, their leaves no larger than a fingernail, growing in clusters on wet rocks or among mosses. Others produce leaves the size of a hand, sprawling across the surface of bogs. The genus Drosera contains nearly 200 species, each adapted to its particular environment, each a variation on the theme of sticky tentacles and patient capture. The mucilage itself is a complex substance. It must be sticky enough to hold struggling prey, yet fluid enough that the plant can reabsorb it after digestion is complete, recycling the material for future captures. It contains digestive enzymes, ready to begin breaking down the insect's proteins and tissues as soon as capture is complete. Some sundews also secrete acids that help dissolve prey, creating a chemical bath that works alongside the enzymes. Watching a sundew capture prey is an exercise in slow-motion drama. The initial contact happens in an instant. Then, the struggle. The insect's wings beating uselessly against the sticky surface, legs pulling free only to stick elsewhere, antennae waving in what might be distress. Gradually, the movements slow. The tentacles, responding to the chemical signals of prey, begin their slow bend inward. Hours later, the leaf is curved around the insect's body, tentacles pressed against it, digestion underway, and then stillness. Days pass. The leaf slowly opens again, revealing only the empty exoskeleton, which will blow away in the wind or wash away in the rain, leaving the sundew ready for its next meal. The movement of sundew tentacles, though slow compared to the Venus flytrap's snap, is still remarkable for a plant. The bending is driven by differential growth on opposite sides of the tentacle. Cells on the outer side of the curve elongate, while cells on the inner side remain unchanged, or even contract slightly. This process, called thigmotropism, is irreversible. Once a tentacle has bent, it cannot unbend the same way. It must grow back straight, a process that takes time and energy. Different species of sundews show different movement strategies. Some species have tentacles only on the edges of leaves, with the center of the leaf remaining flat and covered in sessile digestive glands. When prey is caught on the edge tentacles, the leaf itself curls, bringing the center into contact with the prey. Other species have tentacles covering the entire leaf surface, creating a dense forest of sticky traps. Still others produce long, thin leaves that roll up lengthwise, forming tubes around captured insects. Bladderworts are the fastest traps in the plant kingdom. Among carnivorous plants, the bladderwort holds the speed record. These aquatic or semi-aquatic plants produce tiny bladders, usually less than a few millimeters across, that function as suction traps. Each bladder is a hollow chamber with a hinged door. The plant actively pumps water out of the bladder, creating negative pressure inside. This takes time and energy, but once set, the trap is armed and exquisitely sensitive. Tiny trigger hairs protrude from around the door. When a small organism, perhaps a water flea, mosquito larva, or protozoan, brushes against these hairs, the door springs open. Water rushes into the bladder, carrying the prey with it. The entire event takes between 1 and 5 milliseconds, making it one of the fastest movements in the plant kingdom, even quicker than the Venus flytrap's snap. The door closes immediately behind the prey, sealing it inside, and digestion begins within the bladder. Bladderworts have no roots. They float in ponds, ditches, and wetlands, or grow partially submerged in wet soil. Their leaves are highly divided, creating a feathery appearance that increases surface area for photosynthesis. But scattered among these leaves, often hidden, are hundreds or even thousands of tiny bladders, each one a potential trap. A single plant might capture dozens of prey items per day, processing them in parallel, extracting nutrients that supplement the meager offerings of the nutrient-poor waters they inhabit. The genus Utricularia contains more than 200 species, found on every continent except Antarctica. Some are large and conspicuous, producing bright yellow flowers on tall stalks above the water surface. Others are minuscule, requiring a magnifying glass to appreciate. Despite their small size and delicate appearance, Bladderworts are voracious predators in the microscopic world, shaping the communities of tiny organisms around them. To observe a bladderwort bladder under a microscope is to enter an alien realm. The bladder itself is translucent, revealing the shadowy form of prey items tumbling inside as the water rushed in. The door is a marvel of engineering, a perfect seal when closed, capable of opening in a fraction of a heartbeat. Within, digestive glands line the walls, secreting enzymes that reduce the prey to nutrient-rich fluid. The entire structure exists in a world invisible to the naked eye, yet it is as sophisticated as any animal's trap, as effective as any predator's strategy. The mechanics of the bladderwort trap have been studied extensively, yet they continue to yield surprises. The trigger mechanism is so sensitive that individual protozoa, organisms barely visible under a microscope, can trigger it. The door hinges are flexible yet strong, capable of withstanding the sudden inrush of water without tearing. The seals around the door must be perfect. Even a tiny leak would prevent the negative pressure from building, rendering the trap useless. Recent research has revealed that bladderworts use active ion transport to pump water out of the bladder, creating the negative pressure. This process requires energy in the form of ATP, the universal energy currency of cells. The plant must constantly invest energy to keep its traps armed and ready. But the return on this investment is significant. In nutrient-poor waters where other plants struggle to find nitrogen and phosphorus, bladderworts thrive, their numerous traps providing a steady stream of nutrients. Butterworts use sticky leaves to capture prey. Butterworts, genus Pinguicula, take a simpler approach than Sundews but achieve similar results. Their leaves are broad, flat, and covered with a thin layer of sticky mucilage. Unlike Sundews, butterworts have no mobile tentacles. The leaf itself does not curl. Instead, the entire surface functions as flypaper. Small insects walking across the leaf become mired in the mucus. The more they struggle, the more thoroughly they become coated. Eventually, exhaustion takes them, and they succumb. The name butterwort comes from the greasy, buttery texture of the leaves. This slickness is the mucilage, a complex mix of sugars and proteins that adheres to anything that touches it. Embedded in the leaf surface are thousands of tiny glands. Some produce the mucilage, others secrete digestive enzymes. As the insect struggles and dies, these glands activate, flooding the area around the prey with enzymes that begin breaking down its body. Butterworts are found in temperate and tropical regions, often growing on wet rocks, cliff faces, or in bogs. Their leaves form rosettes close to the ground, often with a pale green or yellowish color that can appear luminous in low light. They produce delicate flowers on tall, thin stalks, flowers that bear a resemblance to violets, with 5 petals and a spurred back. These flowers attract pollinators while the leaves below continue their patient work of trapping and digesting prey. Some species of butterworts have evolved a curious, dual strategy. During the growing season, they produce carnivorous leaves, taking advantage of abundant insect populations to supplement their nutrition. But as winter approaches, they produce a different type of leaf, small, thick, and non-carnivorous, forming a compact rosette that survives the cold. This seasonal shift allows them to balance the energy costs of carnivory against the availability of prey, producing traps only when they are most useful. In traditional use, Butterworts were sometimes employed by farmers to curdle milk, the enzymes on the leaves serving a purpose in cheesemaking. This practical application arose from the same digestive chemistry that allows the plant to break down insect proteins. It is a reminder that the line between the botanical and the chemical, between nature's tools and human ingenuity, is often blurred. The butterwort cares nothing for our uses. It simply exists, catching what it can, surviving where others cannot. The simplicity of the butterwort's trapping mechanism belies its effectiveness. Without moving tentacles or snapping jaws, the butterwort manages to capture plenty of prey. The key is the nature of the mucilage, which is both sticky and somewhat elastic. When an insect lands and tries to walk, its feet stick slightly with each step. The insect pulls free, but each pull requires effort, and each time it touches the leaf again, it sticks anew. Eventually, the accumulated exhaustion from these countless small struggles overwhelms the insect. Some butterworts show a subtle curling response at the edges of leaves when prey is captured, though this movement is much slower and less dramatic than that of sundews. The curling may help ensure that the prey remains in contact with the digestive surface, preventing it from being blown or washed away before digestion is complete. This minimal movement represents a middle ground between the purely passive trapping of some pitcher plants and the more active responses of sundews and Venus flytraps. Cobra lilies stand like hooded serpents. The cobra lily, Darlingtonia californica, is one of the most striking carnivorous plants, native to cold water bogs and seeps in northern California and southern Oregon. It produces tubular leaves that rise from the ground and arch over at the top, forming a hooded structure that resembles a cobra ready to strike. The hood, often veined with red or purple, has a forked, tongue-like appendage hanging from its underside, completing the serpentine illusion. Unlike most pitcher plants, the cobra lily's hood covers the opening of the trap. Insects do not fall directly into the mouth. Instead, they are lured to the hood by nectar and the bright, translucent patches of tissue called fenestrations, which look like exits to the outside world. Confused insects fly or crawl toward these false exits, only to find themselves trapped against the plant's surface. Eventually, exhausted and disoriented, they fall downward into the tubular body of the plant, where downward-pointing hairs prevent escape. At the base of the tube, digestive fluid waits, similar to other pitcher plants. But the cobra lily's fluid is different. It contains no digestive enzymes produced by the plant itself. Instead, the plant relies entirely on bacteria to break down the prey. This is unusual among pitcher plants and suggests that the cobra lily has taken a more passive approach, providing the chamber and relying on microbial partners to do the work of digestion. The cobra lily grows in environments that would challenge most plants. Its roots often dangle in ice-cold spring water, even in summer. It cannot tolerate warm water temperatures and will die if conditions become too hot. This makes it vulnerable to climate change, restricted to a narrow range of habitats where cold, pure water seeps from the ground year-round. In these places, it stands in clusters, dozens of hooded heads rising from the moss and gravel, silent sentinels in a landscape that feels ancient and untouched. To walk among cobra lilies in the wild is to feel transported. They rise from the ground at odd angles, some tall and stately, others short and twisted, their hoods turning to follow some invisible pattern. They are alien in form, yet undeniably beautiful, sculptural and strange. They have inspired myths and legends among those who encounter them. Stories of serpents turned to plant, of guardians watching over hidden springs. Whether such stories hold any truth matters less than the feeling they evoke, that these plants are more than mere vegetation, that they hold some secret, some ancient knowledge written into their form. The cobra lily's reliance on bacterial digestion is an interesting evolutionary strategy. By outsourcing the enzymatic breakdown of prey to bacterial partners, the plant reduces its own metabolic costs. It does not need to produce and secrete digestive enzymes, which requires energy and resources. Instead, it provides a suitable environment for bacteria, which naturally decompose organic material. The plant then absorbs the nutrients released by bacterial action. This strategy may have evolved because cobra lilies grow in such cold environments. Enzyme activity is temperature dependent, and the cold spring water where cobra lilies thrive might slow enzymatic digestion to impractical rates. Bacteria, being living organisms, can regulate their own metabolism and remain active even in cold conditions. By partnering with bacteria, the cobra lily ensures effective digestion despite the challenging thermal environment. Carnivorous plants live in some of the harshest environments on Earth. The places where carnivorous plants thrive are not gentle. Bogs, where water saturates the soil but provides almost no nutrients, the dense mat of peat beneath locking away minerals in a chemical form that roots cannot access. Heathlands, where sandy or rocky soils drain quickly, leaving nothing behind but silica and acidity. Tropical tepuis, table mountains in South America, where rain washes constantly across bare rock, preventing any accumulation of organic material. In these places, conventional plants struggle. Carnivorous plants flourish. The common thread linking these environments is nutrient poverty, particularly nitrogen and phosphorus, elements essential for growth. In rich soils, carnivorous plants cannot compete. Their traps are expensive to produce and maintain, energy that other plants pour into faster growth and larger leaves. But in poor soils, the tables turn. The ability to extract nitrogen from insects becomes a decisive advantage. While other plants starve, carnivorous plants feast on the bodies of flies and ants. This is why carnivorous plants are indicators of ecosystem health. Their presence signals pristine, undisturbed wetlands. When bogs are drained or polluted, when fertilizers run off into wild areas, carnivorous plants disappear. They cannot tolerate enriched soils. The very nutrients that most plants crave poison them, allowing faster-growing competitors to crowd them out. Conservation efforts to protect carnivorous plants are, by extension, efforts to protect entire ecosystems, preserving the delicate balance of poor soils and clean water that these habitats require. Many carnivorous plant habitats are also extraordinarily beautiful. The open expanses of a bog at dawn, mist rising from the surface, sundews glittering with dew, pitcher plants standing like guards, the sound of water trickling through moss, the scent of peat and wildflowers, the sense of isolation and timelessness. These are not wastelands. They are among the most distinctive and precious landscapes on Earth, homes to specialized communities of plants and animals found nowhere else. Bogs in particular represent ecosystems of remarkable constancy. Because they are fed primarily by rainwater rather than groundwater or streams, they remain chemically stable over long periods. The water is acidic, low in nutrients, and low in oxygen. These conditions slow decomposition, causing dead plant material to accumulate as peat rather than breaking down completely. Over millennia, peat deposits can grow meters deep, creating a living archive of past vegetation. Pollen grains preserved in peat layers allow scientists to reconstruct past climates and plant communities, reading the history of the landscape, going back thousands of years. For carnivorous plants, the stability of bogs is crucial. These are plants adapted to specific, unchanging conditions. They cannot quickly adjust to new circumstances. When a bog is disturbed, whether by drainage, pollution, or climate change, the carnivorous plants that depended on its stability often disappear, sometimes within a single generation. This sensitivity makes them valuable indicators of environmental health, but also makes them vulnerable to the rapid changes currently affecting many ecosystems. Some carnivorous plants have evolved symbiotic relationships. While carnivorous plants are predators, some have learned to share their spoils. The fluids within pitcher plants, rich in nutrients from decomposing prey, attract certain insects that have evolved to tolerate or even thrive in this deadly environment. Mosquito larvae, for instance, live within the pitchers of some species, feeding on the bacteria and detritus. They are immune to the digestive enzymes or live in species that lack them. In return, their waste products and remains eventually contribute to the nutrient pool the plant absorbs. Other pitchers host spiders that build webs across the opening, catching insects before they fall in. The spider feeds, and when prey items fall or are dropped into the pitcher, the plant benefits. It is a relationship of convenience rather than true symbiosis, but both parties gain something from the arrangement. The spider gets a hunting ground with a steady supply of insects attracted by the pitcher's lures. The plant gets the dropped or discarded remains. Some pitcher plants have evolved even stranger partnerships. Certain species of Nepenthes in Borneo have pitchers that are used as toilets by tree shrews and bats. The animals perch on the rim or cling to the lid, defecate into the pitcher, and in return receive nectar secreted by the plant. The pitcher receives a steady supply of nitrogen-rich feces, far more reliable than catching insects. This mutualism represents a shift from pure carnivory to a mixed strategy, where the plant still uses a trap structure but captures nutrients from animal waste rather than animal bodies. These relationships challenge our simple categories. Is a plant that receives nutrients from living animals but does not kill them still carnivorous? Or has it evolved beyond carnivory into something else, a partnership where both parties benefit? The answer may depend on how we define the terms, but the reality is that nature rarely respects our neat classifications. Evolution finds solutions that work, and if a pitcher plant can thrive by offering a toilet service to shrews, then that is the path it takes. The mutualistic relationship between Nepenthes species and tree shrews has been studied in detail. The pitchers of these species have modified lids that serve as platforms where shrews can comfortably sit while feeding on nectar. The size and shape of the pitcher opening is perfectly suited to the shrew's anatomy, and the plant produces nectar at peak shrew activity times. This level of coordination suggests a long co-evolutionary history, with plant and animal shaping each other's evolution over countless generations. Similar relationships have been documented with bats. Some Nepenthes species produce pitchers with lids that act as acoustic reflectors, broadcasting the bat's echolocation calls back with unusual clarity. Bats, searching for suitable roosts in the dense forest, are attracted to these acoustic beacons. They investigate, find the pitcher, and use it as a toilet in exchange for nectar. The plant benefits from bat droppings, which are rich in nitrogen from the insects the bats have eaten. It is a three-way transfer of nutrients: insects to bat, bat to plant, plant to bat, in the form of nectar energy. Carnivorous plants produce beautiful and fragrant flowers. Despite their fearsome reputation, or perhaps because of it, Carnivorous plants produce some of the most delicate and beautiful flowers in the botanical world. Venus flytraps bloom in late spring, sending up a tall stalk crowned with white five-petaled flowers. Pitcher plants produce large, nodding blooms, often pink, purple, or deep red, with complex structures that attract specific pollinators. Sundews lift their flowers high above the leaves on wiry stems, keeping pollinators safe from the sticky traps below. This separation of flowers from traps is no accident. Carnivorous plants need pollinators to reproduce, and killing the very insects that carry their pollen would be counterproductive. By elevating flowers well above the trapping leaves, by using different scents and colors that attract pollinators, rather than prey, the plants ensure that the insects they need for reproduction do not become meals. It is a delicate balance, managing two contradictory needs, attracting some insects to die, others to live and carry pollen. The flowers themselves are often remarkably beautiful. Butterworts produce blooms that resemble tiny orchids or violets. Bladderworts send up spikes of bright yellow or purple flowers that belie the tiny hidden traps below. Some pitcher plants have flowers so large and showy they are cultivated purely for ornamental purposes, their carnivorous nature almost an afterthought. The contrast is striking. Below, death and digestion. Above, color and fragrance, and the promise of life continuing. This duality makes carnivorous plants endlessly fascinating. They are not mere killers. They are participants in the full cycle of life, taking what they need to survive, while also giving back through pollination and seed production. Their flowers attract bees, butterflies, moths, and flies, insects that feed without fear, sipping nectar and gathering pollen. In this way, carnivorous plants are generous even as they are deadly, offering sustenance to some while consuming others. The timing of flowering is also carefully coordinated. Many carnivorous plants bloom before they produce their main flush of traps, or they bloom early in the season when pollinator populations are building, but prey insects are still relatively scarce. This temporal separation provides an additional safeguard, reducing the chance that a pollinator visiting a flower will subsequently wander down to the traps below and become a meal instead of a reproductive partner. The pollination strategies of carnivorous plants are as diverse as their trapping mechanisms. Some are generalists, accepting pollen transfer from any visiting insect. Others have specialized relationships with particular pollinators. Certain pitcher plants have flowers with complex architectures that only large bees can effectively navigate, ensuring that pollination is performed by insects too large and strong to be easily captured by the traps. The co-evolution between carnivorous plants and their pollinators represents millions of years of refinement, each species shaping the other's form and behavior. Carnivorous plants are threatened worldwide by habitat loss. As human populations expand and wild lands shrink, carnivorous plants face increasing threats. Wetlands are drained for agriculture or development. Bogs are mined for peat, their centuries-old accumulations stripped away in years. Pollution, particularly nutrient runoff from farms and cities, enriches the poor soils that carnivorous plants require, making them unsuitable. Collectors, attracted by the plant's exotic nature, harvest them from the wild, sometimes driving local populations to extinction. Many species of carnivorous plants are now rare or endangered. Some exist only in a handful of locations, their total wild populations numbering in the hundreds or fewer. The Venus flytrap, despite being iconic and widely cultivated, is endangered in the wild. Its native range restricted to a small area of the Carolinas where development pressure is intense. Certain species of pitcher plants are so rare that their exact locations are kept secret to prevent poaching. Conservation efforts are underway, Botanical gardens and specialized nurseries propagate carnivorous plants, preserving genetic diversity and providing plants for sale to reduce pressure on wild populations. Protected areas have been established around critical habitats. Restoration projects aim to rehabilitate degraded bogs and wetlands, replanting native carnivorous plants and recreating the conditions they need to survive. But the challenge is immense. Carnivorous plants require very specific conditions that are increasingly hard to maintain in a changing world. Climate change alters rainfall patterns and temperature regimes. Rising sea levels threaten coastal bog habitats. Fire suppression changes the ecology of habitats that evolved with regular burning. Each loss is not just a species declining, but an entire ecosystem unraveling, the intricate web of relationships that sustains these places fraying thread by thread. The illegal collection of carnivorous plants remains a significant problem. Despite legal protections in many jurisdictions, poachers continue to harvest wild plants for sale to collectors. The rarest species command high prices, creating economic incentives for theft. Venus flytraps, in particular, have been heavily poached from their native habitats, with some commercial collectors removing thousands of plants. Law enforcement efforts have increased, but the remote nature of many carnivorous plant habitats makes policing difficult. Habitat fragmentation compounds these problems. When a large bog is divided into smaller patches by roads, farms, or development, the resulting fragments may be too small to sustain viable populations of carnivorous plants. Small populations are vulnerable to genetic bottlenecks, where inbreeding reduces fitness and adaptability. They are also more susceptible to local extinction from random events like fires, floods, or disease outbreaks. Without corridors connecting habitat patches, recolonization after local extinction becomes impossible. You can grow carnivorous plants at home with the right conditions. Despite their exotic nature, many carnivorous plants are surprisingly easy to grow at home if their needs are met. The key is understanding that they do not want fertilizer, rich soil, or tap water. They want poor conditions, the same nutrient poverty that makes them carnivorous in the first place. This means planting them in a mix of sphagnum, moss, and sand, never in potting soil. It means watering them with distilled water or rainwater, never tap water, which contains minerals that will poison them over time. Venus flytraps, perhaps the most popular carnivorous plant, are relatively forgiving if given full sun and kept moist. They require a winter dormancy, a period of cold rest, but can be grown outdoors in many temperate climates. Sundews are even easier. Many species thriving on windowsills, with bright light and a saucer of distilled water to keep their roots wet. Pitcher plants, particularly the North American Sarracenia, can be grown outdoors in bogs or container gardens. Their tall pitchers adding vertical interest to a garden. Growing carnivorous plants is a lesson in patience and observation. They do not grow quickly. Traps take time to develop. A Venus flytrap might produce only a few new leaves each month. Pitcher plants grow a flush of pitchers in spring, then rest, then perhaps produce another flush in fall. Feeding them is unnecessary in most cases. If grown outdoors, they will catch their own prey. Indoors, an occasional small insect can be offered, but overfeeding is harmful, causing traps to rot. The reward for this patience is witnessing these plants in action. Watching a Venus flytrap snap shut on a fly never gets old. Seeing a sundew's tentacles slowly curl around a gnat is mesmerizing. Discovering that a pitcher has captured a moth, the wings visible through the translucent walls, connects you to the plant's primal purpose. These are not passive houseplants. They are hunters, and sharing space with them brings a small, thrilling piece of the wild into your home. One of the most common mistakes new growers make is trying to help the plants by providing them with fertilizer or rich soil. This seems logical from the perspective of conventional gardening, where the goal is to provide plants with as many nutrients as possible. But for carnivorous plants, this approach is fatal. Their roots are adapted to nutrient-poor conditions and cannot handle the salt concentrations found in fertilizers or the mineral content of regular potting soil. Within weeks or months, the plants will show signs of distress and eventually die. Another common error is insufficient light. Most carnivorous plants require very bright light, equivalent to full sun or nearly so. A north-facing window is usually insufficient. The plants may survive in low light, but will grow poorly, producing small traps and failing to develop the vibrant colors that many species display when properly grown. For indoor cultivation, supplemental artificial lighting is often necessary, particularly during winter months when natural daylight is limited. Carnivorous plant traps can be grouped into 5 basic types. Across the diversity of carnivorous plants, 5 fundamental trap mechanisms emerge: pitfall traps, flypaper traps, snap traps, bladder traps, and lobster pot traps. Each represents a different solution to the problem of capturing prey, and each has evolved independently in different plant lineages. Understanding these categories helps organize the overwhelming variety of carnivorous species into comprehensible groups. Pitfall traps, seen in pitcher plants, rely on gravity. Prey falls into a chamber from which escape is impossible due to slippery walls, downward-pointing hairs, or drowning in fluid. Flypaper traps, like those of Sundews and Butterworts, use sticky mucilage to ensnare prey on the leaf surface. Snap traps, unique to Venus flytraps and closely related species, use rapid movement to close around prey. Bladder traps, found only in Bladderworts, use suction to pull prey into a sealed chamber. Lobster pot traps, seen in corkscrew plants, use inward-pointing hairs to guide prey deeper into a tube, making retreat impossible. Some plants use variations or combinations of these mechanisms. Certain pitcher plants combine pitfall traps with features of lobster pots, using hairs to guide insects downward. Some sundews have leaves that curl, adding an element of snap trap to their flypaper approach. Evolution is not constrained by our categories, and plants blend strategies when it offers an advantage. What unites all these traps is their purpose and their cost. They all capture prey to extract nutrients, and they all require energy to produce and maintain. This energy investment is only worthwhile in nutrient-poor habitats. In rich soils, the cost exceeds the benefit, and carnivorous plants cannot compete. But in their specialized niches, these traps make the difference between thriving and perishing, between tapping an alternative food source and slowly starving in a land of plenty where everything is abundant except the nutrients that matter most. The evolution of each trap type involved solving multiple engineering challenges simultaneously. A pitfall trap must be deep enough to prevent escape, but not so deep that the plant cannot absorb nutrients from prey at the bottom. The walls must be slippery, but the structure must also be strong enough to support its own weight when filled with water. Flypaper traps must produce mucilage, that is, sticky, but not so viscous that it cannot be reabsorbed. Snap traps must move quickly, but also avoid false triggers that would waste energy. Each solution represents a delicate balance of competing requirements. The rarity of some trap types reflects their difficulty of evolution. Snap traps are found in only a handful of closely related species, all in the genus Dionea and its close relatives. This suggests that the snap mechanism is difficult to evolve, requiring multiple coordinated mutations that must all be present simultaneously for the trap to function. Bladder traps are similarly restricted, found only in the genus Utricularia. In contrast, pitfall traps and flypaper traps have evolved multiple times independently, suggesting that these mechanisms are more readily accessible to evolutionary innovation. The digestive enzymes of carnivorous plants are similar to those in animal stomachs. One of the most surprising aspects of carnivorous plants is the chemistry of digestion. The enzymes they produce are remarkably similar to those found in the stomachs and intestines of animals. Proteases break down proteins into amino acids. Phosphatases release phosphorus from organic molecules. Chitinases dissolve the chitin of insect exoskeletons. Even the pH of the digestive fluid, often acidic, mirrors the gastric juices of predatory animals. This similarity is not coincidence, but convergence. The challenge of digesting prey is the same whether you are a plant or an animal. Proteins must be broken down, nutrients extracted, waste left behind. Evolution has arrived at similar chemical solutions, producing enzymes that serve the same functions. In some cases, the genes that produce these enzymes in carnivorous plants are related to genes in their non-carnivorous ancestors that served different purposes, repurposed through mutation and selection into tools of digestion. Not all carnivorous plants produce their own enzymes. Some, like the cobra lily, rely entirely on bacteria living within their pitchers to do the work. Others use a mix, producing some enzymes while hosting bacterial communities that contribute others. This reliance on bacteria is a form of outsourcing, reducing the plant's energy expenditure while still achieving the same result. Prey broken down, nutrients absorbed. The presence of these digestive mechanisms is one of the defining features of carnivory. Many plants can trap insects incidentally. Sticky stems, tight flower structures, even certain hairs can catch small insects. But without the ability to digest and absorb nutrients from those insects, the plant gains nothing. True carnivorous plants go beyond trapping to extraction, actively reclaiming the nitrogen, phosphorus, and other elements locked within insect bodies and making them part of their own tissues. Recent genetic studies have revealed how carnivorous plants acquired their digestive enzymes. In many cases, genes that originally served defensive functions in the fulbrines, plants' non-carnivorous ancestors, were duplicated and modified. For example, proteases that once protected the plant from herbivores by breaking down the digestive enzymes of attacking insects were repurposed to digest the insects themselves. This reuse of existing genetic material is a common pattern in evolution, where new functions emerge from the modification of old ones, rather than arising entirely de novo. The regulation of enzyme production is also sophisticated. Carnivorous plants do not produce digestive enzymes constantly. This would be wastefully expensive. Instead, enzyme production is triggered by the presence of prey. Chemical signals from the struggling insect, or from the breakdown products of its cuticle, stimulate the glands to begin secreting enzymes. This conditional response ensures that resources are invested in digestion only when there is something to digest. Some carnivorous plants can survive years without catching prey. While carnivorous plants benefit enormously from prey capture, they are not entirely dependent on it. As long as they receive adequate light for photosynthesis, they can survive for extended periods without catching anything. Studies have shown that carnivorous plants grown without prey grow more slowly, produce fewer traps, and may not flower, but they do not immediately die. This resilience is important in the wild, where insect populations fluctuate seasonally. During winter, or in particularly wet seasons, prey may be scarce. The plants enter a period of dormancy or slow growth, relying on stored nutrients and the energy they can generate through photosynthesis. When conditions improve and prey becomes abundant again, they resume active trapping and grow more vigorously. The trade-off between carnivory and pure photosynthesis is a sliding scale. In times of plenty, catching insects provides a significant boost, allowing faster growth and more reproduction. In lean times, the plants fall back on their photosynthetic capabilities, becoming more like conventional plants. This flexibility is part of their success, allowing them to exploit carnivory when it is advantageous, while not being locked into a strategy that would be fatal if prey disappeared. For growers, this means that carnivorous plants will not die if they fail to catch insects regularly. An indoor Venus flytrap that catches nothing for months will survive, though it may look less robust than one that feeds regularly. This resilience makes them more forgiving as houseplants than their reputation suggests. They are specialist plants, yes, requiring specific conditions, but within those parameters, they are remarkably adaptable. The proportion of nitrogen that carnivorous plants derive from prey versus from their roots varies by species and environmental conditions. Some studies have found that carnivorous plants in the wild obtain as much as 80% of their nitrogen from insects, while others rely on insects for only 20–40% of their needs. The variation depends on factors like soil nitrogen availability, insect abundance, and the efficiency of the plant's traps. Even within a single species, different populations may show different degrees of reliance on carnivory. This variability has important implications for conservation. It suggests that carnivorous plants might be able to persist in habitats where insect populations have declined due to pesticides or other factors, at least for a time. However, their growth and reproduction would likely be impaired, potentially leading to long-term population declines, even if the plants do not immediately disappear. Protecting carnivorous plants, therefore, requires protecting not just their habitats, but also the insect communities they depend on. Ancient cultures knew about carnivorous plants long before science. The Venus flytrap was first formally described to European science in the 1760s, causing a sensation. How could a plant move and consume animals? It challenged prevailing assumptions about the natural order. But indigenous peoples in the regions where these plants grew had known them for far longer, incorporating them into stories and medicinal practices. Pitcher plants appear in various cultural traditions. Some indigenous North American tribes used pitcher plant roots for medicinal purposes, treating ailments ranging from digestive issues to smallpox. Whether these treatments were effective is uncertain, but the recognition of these plants as unusual and powerful is clear. In Southeast Asia, where giant pitcher plants grow, local peoples have long known of them, sometimes using the pitchers as containers for cooking rice or carrying water. The challenge for early botanists was categorization. Plants were supposed to be passive, rooted, drawing sustenance from soil and sun. Here were organisms that actively trapped and digested animals. Were they plants or something else? Early descriptions struggled with this. Some writers speculating that carnivorous plants represented a bridge between the plant and animal kingdoms, a primitive form of animal-plant hybrid. It was Charles Darwin who finally put carnivorous plants on solid scientific footing. His book, Insectivorous Plants, published in 1875, detailed exhaustive experiments on sundews, demonstrating beyond doubt that they digested prey and absorbed nutrients. Darwin fed sundews various substances, measured their responses, timed the movement of tentacles, and documented everything with meticulous care. His work established that carnivorous plants were indeed plants, but plants that had evolved a remarkable strategy for survival. Darwin's experiments were groundbreaking in their rigor and creativity. He tested sundews with everything from raw meat to gelatin to glass beads, determining what triggered the plant's response and what did not. He measured the bending of individual tentacles with a microscope, tracking their movement over hours and days. He even tested whether sundews could distinguish between nutritious and non-nutritious substances, finding that they responded more strongly to items containing nitrogen. Darwin's fascination with carnivorous plants was part of his broader interest in how plants respond to their environments. He saw in these plants evidence of something approaching behavior, a responsiveness that blurred the line between the sessile world of plants and the active world of animals. His work on carnivorous plants complemented his studies of climbing plants, phototropism, and other plant movements, all of which demonstrated that plants were far more dynamic and responsive than commonly believed. Carnivorous plants inspired early science fiction and horror. The idea of a plant that eats animals captured the Victorian imagination. Shortly after carnivorous plants became widely known, stories began appearing of man-eating plants, monstrous versions of the Venus flytrap or pitcher plant, large enough to consume humans. These tales, often set in exotic jungles or on distant planets, played on fears of the unknown and reversed the expected roles of predator and prey. One famous hoax involved the man-eating tree of Madagascar, described in sensational newspaper accounts in the 1,870 seconds. The story claimed that a tribe in Madagascar used a tree with tentacle-like branches to sacrifice victims, the tree squeezing them to death and consuming their blood. The account was entirely fabricated, but it was widely believed and reprinted, feeding public fascination with carnivorous plants. These fictional plants influenced science fiction for generations. In H.G. Wells' short stories, in pulp magazines of the early 20th century, in films and comic books, oversized carnivorous plants became stock villains. They represented nature turning against humanity, the domesticated and safe world of plants revealing hidden savagery. Even today, carnivorous plants appear in popular culture as monsters or threats, from the musical Little Shop of Horrors to video games featuring giant flytraps as enemies. The real carnivorous plants, of course, pose no threat to humans. The largest could potentially capture a very small mammal, but nothing approaching human size. Yet the psychological impact remains. These plants remind us that nature contains surprises, that the familiar can suddenly reveal itself as strange, that the world is more complex and unsettling than our simplified categories suggest. In their small way, carnivorous plants challenge our place in the natural order, showing us that predation is not the sole domain of animals. The persistence of carnivorous plants in popular culture reflects something deeper than mere entertainment. They tap into primal anxieties about the natural world. For most of human history, nature was dangerous, something to be feared and controlled. The development of agriculture and civilization created the illusion of human dominance over nature. With plants as passive resources to be cultivated and harvested, carnivorous plants subvert this narrative. They are plants that fight back, that consume rather than being consumed. This reversal is both fascinating and unsettling, a reminder that nature retains agency and that our control is never as complete as we imagine. Fossil evidence shows carnivorous plants are millions of years old. The fossil record for carnivorous plants is sparse. Soft tissues rarely fossilize well, and traps, being modified leaves, decompose quickly. but evidence exists that carnivorous plants have been around for millions of years. Pollen grains from ancient carnivorous plants have been found in amber, trapped alongside the insects that likely would have been their prey. Fossilized leaves bearing resemblance to modern pitcher plants and sundews have been discovered in rock formations dating back millions of years. The most compelling evidence comes from genetic analysis. By comparing the DNA of modern, carnivorous plants with their non-carnivorous relatives, scientists can estimate when the lineages diverged. These molecular clocks suggest that carnivory evolved independently in different plant families tens of millions of years ago, perhaps triggered by the expansion of nutrient-poor habitats during periods of global climate change. During the age of dinosaurs, carnivorous plants may have dotted the landscapes just as they do today. They're traps, waiting for insects that looked very different from modern species, but filled the same ecological roles. Throughout the rise and fall of species, through ice ages and warm periods, through continental drift and the evolution of flowering plants, carnivorous plants persisted, their specialized adaptations allowing them to survive in niches where few others could thrive. This longevity speaks to the success of carnivory as a strategy. It is not a dead end or a novelty. It is a viable, long-term solution to the problem of nutrient poverty, one that has sustained entire lineages of plants across geological time. The Venus flytrap you might grow on your windowsill is the inheritor of a strategy millions of years in the making, refined through countless generations, a living link to deep time. The fossil record, though incomplete, provides tantalizing glimpses of ancient carnivorous plants. Fossilized seeds resembling those of modern Aldrovanda, the waterwheel plant related to Venus flytraps, have been found in European deposits from the Tertiary period, tens of millions of years ago. This suggests that snap traps, now represented by only a handful of living species, were once more diverse. Similarly, fossilized pollen and seeds attributed to ancient Droseraceae have been found in various locations, indicating that sundews and their relatives have a long history. The rarity of carnivorous plant fossils may itself tell us something about these plants. They typically grow in acidic, waterlogged environments that are not conducive to fossilization. The very conditions that favor carnivorous plants in life work against their preservation after death. This creates a systematic bias in the fossil record, likely hiding much of the true history of these lineages. The mechanisms behind trap movement are still being discovered. Despite more than a century of study, scientists continue to uncover new details about how carnivorous plants work. The Venus flytrap's snap, for instance, has only recently been fully explained at the cellular level. Advances in high-speed imaging, genetic analysis, and modeling have revealed that the trap's closure involves rapid changes in cell curvature, driven by differential growth and hydraulic pressure, a mechanism far more complex than early researchers imagined. Similarly, the counting mechanism that prevents false triggers in the Venus flytrap has been traced to electrical signals similar to those in animal nerves. When a trigger hair is bent, it generates an electrical pulse that travels through the trap. If a second pulse arrives within about 20 seconds, the trap closes. This electrical memory, a form of short-term information storage in a brainless organism, has implications beyond botany, informing our understanding of how simple systems can process information. Sundew movement, once thought to be purely hydraulic, has been found to involve both water movement and irreversible cell growth. The tentacles don't just bend through pressure changes, they actually grow differentially on one side, bending permanently toward prey. After digestion, those tentacles straighten through growth on the opposite side. It is a slow, metabolically expensive process. but it ensures maximum contact between digestive surfaces and prey. Each discovery raises new questions. How do plants sense prey chemically, distinguishing between food and debris? What evolutionary pressures led to such sophisticated mechanisms? How much variation exists between individuals, and could artificial selection breed even more effective traps? As technology advances, allowing researchers to peer deeper into the molecular and physical processes of these plants, the answers become clearer, but the fundamental wonder remains. How does a plant without a brain, without nerves, without any central coordination, accomplish such complex behaviors? Recent work on Venus flytraps has revealed the molecular basis of their electrical signaling. The trigger hairs contain mechanosensitive ion channels that open when the hair is bent, allowing charged ions to flow across cell membranes. This creates an electrical potential change that propagates through the trap tissue. The trap appears to integrate these signals, somehow counting them and triggering closure only when the threshold is reached. The molecular mechanisms underlying this counting ability remain partially mysterious, but they involve changes in calcium ion concentrations and the activation of specific genes. Understanding these mechanisms has potential applications in robotics and materials science, where researchers are interested in creating materials that can sense and respond to mechanical stimuli without electronic controls. Carnivorous plants teach us about evolution and adaptation. Beyond their intrinsic fascination, carnivorous plants serve as powerful examples for understanding evolution. They demonstrate convergent evolution beautifully, showing how similar environmental pressures lead to similar solutions in unrelated lineages. Pitcher plants evolved independently in Asia, North America, South America, and Australia, yet all arrived at variations of the same basic design: a modified leaf forming a trap filled with digestive fluid. They also illustrate the concept of evolutionary trade-offs. Carnivorous plants invest heavily in traps at the expense of other traits. Their root systems are often reduced, their growth rates slower than non-carnivorous plants in rich soil. These trade-offs make sense only in specific contexts. Carnivory is not universally beneficial. It is beneficial only when and where the costs are outweighed by the gains. This context dependency is fundamental to understanding evolution, which produces not perfect organisms, but organisms well suited to their particular circumstances. Carnivorous plants also show us that evolution is opportunistic and creative. The traps of carnivorous plants are modified versions of ordinary plant structures. Leaves became pitchers, tentacles, jaws. Glands that once secreted nectar now produce digestive enzymes. Hairs that guided water now trigger traps. Evolution works with what is available, repurposing existing structures into new functions through gradual modification. Each step along the way must be functional, must offer some advantage, no matter how small. For students of biology, carnivorous plants are perfect teaching tools. They are tangible, observable, and dramatic. They make evolution concrete. Watching a Venus flytrap close is not just seeing a cool plant trick. It is witnessing millions of years of natural selection written into living tissue, responding in real time to the presence of prey. Every carnivorous plant is a reminder that life is endlessly inventive, that adaptation can take forms we never imagined, and that even the most unlikely strategies can succeed if they solve the right problem in the right place. The study of carnivorous plants has contributed significantly to our understanding of how complex traits evolve. One key insight is that complexity can arise gradually through the accumulation of small changes, each beneficial on its own. A slightly sticky leaf helps the plant capture some nutrients from decomposing insects. A leaf that produces a small amount of digestive enzyme captures slightly more nutrients. A leaf that curves slightly when touched keeps prey in contact with digestive surfaces slightly longer. None of these changes alone creates a fully functional carnivorous plant, but each provides some benefit, allowing natural selection to favor it. Over many generations, these small improvements accumulate, and what began as a barely detectable tendency becomes a sophisticated trap. Hybridization creates stunning new varieties. In cultivation, carnivorous plants can be hybridized, creating new varieties that combine traits from different species. Pitcher plant enthusiasts, in particular, have created hundreds of hybrids, crossing North American Sarracenia species to produce pitchers with unusual colors, patterns, and shapes. Some hybrids show red veining on white backgrounds. Others display gradients from green to deep purple. The flowers, too, can be altered, producing blooms of uncommon hues. These hybrids are not found in nature. Most carnivorous plants are geographically isolated from related species, preventing natural hybridization. But in cultivation, where species from different regions can be grown side by side, hybridization becomes possible. Breeders hand-pollinate flowers, transferring pollen from one species to another, then grow out the seeds to see what emerges. The results are often striking, combining the best features of both parents or creating entirely new forms. Hybridization has also helped conservation efforts. Some critically endangered carnivorous plants exist in such small numbers that genetic diversity is low, leading to inbreeding depression. Carefully managed breeding programs, including selective hybridization with closely related species, can introduce new genetic variation, strengthening populations that might otherwise decline. Seedlings from these programs can then be used in restoration projects, replanting habitats where carnivorous plants have disappeared. For hobbyists, hybrids offer aesthetic variety and the challenge of cultivation. Some hybrids are more vigorous than their parent species, growing larger or producing more traps. Others are more tolerant of temperature fluctuations or varying light levels, making them easier to keep alive in less than ideal conditions. The world of carnivorous plant cultivation has become a community of experimentation and sharing, with growers around the globe exchanging seeds, cuttings, and knowledge about these remarkable organisms. The genetics of carnivorous plant hybridization reveals interesting patterns. Many carnivorous plants have chromosome numbers that differ between species, which can create challenges for hybridization. When species with different chromosome numbers are crossed, the resulting hybrids may be sterile, unable to produce viable seeds of their own. However, these sterile hybrids can still be propagated vegetatively through division or cuttings, allowing them to persist in cultivation even if they cannot reproduce sexually. Some hybrid combinations are more successful than others. Closely related species typically hybridize easily and produce vigorous, fertile offspring. More distant crosses may produce weak or deformed plants, or may fail entirely. Breeders learn through trial and error which combinations work, gradually building up knowledge of which genetic backgrounds are compatible. This experimentation has produced a vast array of cultivated forms, far exceeding the diversity found in nature. Some carnivorous plants are surprisingly tiny. When people think of carnivorous plants, they often imagine the large pitchers of tropical species or the hand-sized rosettes of Venus flytraps. But many carnivorous plants are diminutive, requiring a magnifying glass to appreciate fully. Some sundews have leaves no larger than a fingernail. Certain bladderworts are entirely microscopic in their trapping structures, the bladders measuring fractions of a millimeter. These miniature carnivores live in specialized micro-habitats, crevices in rocks, mats of moss, the water films on wet surfaces. They capture prey appropriate to their size: mites, springtails, protozoa, nematodes. Their small size is not a limitation but an adaptation, allowing them to thrive in spaces where larger plants cannot establish themselves. A crack in a cliff face, perpetually moist from seepage, might host a population of tiny sundews, each one a complete carnivorous plant, operating at a Lilliputian scale. Growing miniature carnivorous plants requires careful attention. They dry out quickly, their small size offering little buffer against environmental changes. Yet, they can be enchanting in terrariums, where conditions can be controlled precisely. A small glass container, filled with moss and kept humid, can become a miniature bog ecosystem, complete with tiny carnivorous plants capturing prey invisible to the naked eye. The existence of these tiny species reminds us that carnivory scales across size ranges. The principles are the same whether the plant is capturing elephants of the insect world or barely visible microorganisms. A trap is a trap, digestion is digestion, and nutrient extraction is nutrient extraction, regardless of scale. In this, carnivorous plants demonstrate that biological strategies can be implemented at vastly different sizes, each successful within its own context. Some of the smallest carnivorous plants show remarkable adaptations to their microscopic world. Tiny bladderworts that grow in films of water on wet rocks have bladders so small that they capture single-celled organisms like paramecia and rotifers. These bladders are no larger than the width of a human hair, yet they contain all the functional components of larger bladders: the door, the trigger hairs, the digestive glands. The scaling down of these structures to such tiny dimensions represents an engineering marvel. Miniature sundews face different challenges than their larger relatives. Their tiny size means they have very little water storage capacity, making them vulnerable to desiccation. Many grow in constantly moist microhabitats, where water is always available. Their tentacles are proportionally shorter and the mucilage droplets smaller, but the trapping mechanism remains effective at their scale. A gnat that would be a small meal for a large sundew is a substantial capture for a miniature species. Carnivorous plants play unique roles in their ecosystems. Beyond their individual survival, carnivorous plants influence the ecosystems around them. By capturing and digesting insects, they alter nutrient flows. In nutrient-poor bogs, where nitrogen is scarce, the nitrogen brought into the system through insect capture by carnivorous plants eventually returns to the environment when the plants die and decompose. This represents a transfer of nutrients from the terrestrial insect population into the bog ecosystem, a small but significant input. Carnivorous plants also provide habitat. Pitcher plants, with their water-filled chambers, are miniature ecosystems. As mentioned earlier, many insects and other organisms live within or around pitcher plants, forming complex food webs. Mosquito larvae, midges, spiders, mites, and even bacteria and protozoans inhabit the pitchers, creating communities that are found nowhere else. Researchers studying these communities have discovered new species and documented intricate relationships between inhabitants. The flowers of carnivorous plants, elevated above the traps, serve as nectar sources for pollinators. In spring and summer, pitcher plant flowers attract bees, flies, and beetles, providing food when other flowers may be scarce in the harsh bog environment. This makes carnivorous plants keystone species in some habitats, supporting biodiversity through their role as both predator and resource. When carnivorous plant populations decline, the effects ripple outward. Insect populations may change without the predation pressure. Pollinators lose a food source. The unique communities within pitchers disappear. the slow reintroduction of nutrients into the ecosystem slows. Protecting carnivorous plants is thus about more than saving a single species. It is about preserving the intricate web of relationships that make these ecosystems function. The role of carnivorous plants in nutrient cycling has been quantified in some ecosystems. Studies have shown that in certain bogs, carnivorous plants can capture a significant percentage of the total insect biomass present. This insect biomass, once digested, contributes nitrogen and phosphorus to the plants, which eventually release these nutrients back into the ecosystem when leaves die and decompose, or when the plants themselves die. This nutrient input can be particularly important in bogs, which are otherwise extremely nutrient poor. The nitrogen captured from insects may represent one of the few external inputs of nutrients to these closed systems. Without carnivorous plants performing this function, bog ecosystems might be even more nutrient limited, supporting less biological diversity overall. In this way, carnivorous plants can be seen as ecosystem engineers, modifying their environment in ways that affect many other species. The colors of carnivorous plants serve multiple functions. Carnivorous plants display a stunning array of colors: deep reds, vibrant greens, purples, yellows, even whites with intricate veining. These colors are not merely decorative. They serve functions related to prey capture and plant health. Red pigments, called anthocyanins, protect tissues from intense sunlight, a concern in open bogs and heathlands where shade is minimal. They also attract certain insects, particularly those that associate red with nectar or carrion. The translucent windows, or fenestrations, found in some pitcher plants, create false exits that confuse trapped prey. Sunlight filtering through these windows makes them appear as openings to the outside world. Insects fly or crawl toward the light, only to find solid leaf tissue, exhausting themselves in futile escape attempts. The pattern of these windows, their size and placement, has been refined through evolution to maximize disorientation. Contrasting colors guide insects toward traps. A pitcher plant might have a green body with a red rim, drawing the eye and the insect toward the opening. Nectar guides, lines of color or texture, lead from the outer edges of a trap toward its center, much like the nectar guides on ordinary flowers lead pollinators to pollen and nectar. But in carnivorous plants, these guides lead toward death. Some carnivorous plants change color seasonally or in response to light conditions. A Venus flytrap grown in bright sun develops deep red interiors to its traps, while one grown in shade remains green. This plasticity allows the plant to optimize both photosynthesis and prey attraction depending on conditions. The colors of carnivorous plants are thus functional art, beauty that serves survival, aesthetics shaped by the relentless pressure of natural selection. The biochemistry of carnivorous plant coloration is complex. Anthocyanins, the red and purple pigments, are produced through a metabolic pathway that requires significant energy investment. The plant must balance the benefits of these pigments: UV protection, prey attraction, and possibly other functions against their costs. In high light conditions, where UV damage is a significant risk, the benefits clearly outweigh the costs, and plants produce abundant anthocyanins. Interestingly, some carnivorous plants produce their most vivid colors when nutrient-stressed. This might seem counterintuitive, but it makes sense from an adaptive perspective. When nitrogen is scarce, the plant needs to catch more prey, and more vivid coloration may help attract insects. At the same time, the plant has less nitrogen available for chlorophyll production, so the green color fades, making the red and purple pigments more visible. This creates a feedback loop where nutrient stress leads to better prey attraction, potentially alleviating the stress. Carnivorous plants require specific water quality. One of the most critical factors in growing carnivorous plants, and one of the most common reasons for failure, is water quality. Carnivorous plants evolved in environments where water is pure, whether from rain, snowmelt, or springs filtering through rock. This water contains almost no dissolved minerals. The plant's roots and tissues are adapted to this purity and cannot tolerate the minerals found in tap water. Tap water typically contains calcium, magnesium, chlorine, fluoride, and other dissolved substances. These minerals accumulate in the soil over time, building up to toxic levels. The plants show signs of stress, browning leaf tips, reduced growth, traps that fail to open or close properly. Eventually, the plant dies, poisoned by what would nourish ordinary plants. This is why carnivorous plants are often recommended for distilled water, reverse osmosis water, or collected rainwater. In their natural habitats, carnivorous plants receive constant flushing by rain or flowing water, preventing mineral buildup. In cultivation, growers replicate this by using pure water and ensuring good drainage. Some growers use the tray method, placing pots in shallow trays of water so the plants can wick up moisture as needed, changing the water regularly to prevent stagnation. Understanding the water requirements of carnivorous plants highlights their specialization. They have traded the ability to extract minerals from water and soil for the ability to extract nutrients from prey. This is an exclusive trade. They cannot do both effectively. It is a reminder that evolutionary adaptation is not about becoming good at everything, but about becoming excellent at one specific strategy, even if that strategy imposes strict limitations. The mechanism behind the sensitivity to minerals involves the structure of carnivorous plant roots. Unlike most plants, which have extensive root systems with specialized structures for selective uptake of nutrients, carnivorous plant roots are often reduced and simplified. They function primarily for water uptake and anchoring, not for nutrient acquisition. The cells in these roots lack many of the transporters and mechanisms that would allow them to regulate mineral uptake, making them vulnerable to mineral toxicity. When minerals accumulate in the growing medium, they create an osmotic imbalance. The high concentration of dissolved substances outside the roots makes it difficult for the plant to take up water. At the same time, some minerals can enter the plant passively and accumulate to toxic levels. Calcium, for example, can interfere with cellular processes when present in excess. The result is a plant that is simultaneously drought-stressed, unable to take up enough water, and poisoned, accumulating toxic minerals. Temperature requirements vary widely among species. Carnivorous plants come from diverse climates, and their temperature requirements reflect this diversity. Tropical pitcher plants from Southeast Asian rainforests require warm conditions year-round, with high humidity and no frost. Temperate species, like the Venus flytrap and most North American pitcher plants, require a cold winter dormancy period, exposure to temperatures near or below freezing for several months. Without this cold period, they weaken and eventually die. Some carnivorous plants come from highland tropics, where nights are cool even though days are warm. These species need temperature fluctuation, growing poorly in constant warmth. Others, from Mediterranean climates, tolerate dry summers and wet winters, going dormant during heat rather than cold. Understanding a carnivorous plant's origin is essential to providing the right conditions in cultivation. The need for winter dormancy in temperate species surprises many growers. It seems counterintuitive that a plant would need to be cold to stay healthy. But for these species, the cold period triggers essential physiological processes. It resets growth cycles, allows the plant to rest and conserve energy, and prepares it for vigorous spring growth. Without dormancy, the plant exhausts itself, producing smaller and fewer traps each year until it dies. Providing dormancy can be as simple as moving a potted plant to an unheated garage or leaving it outdoors in a sheltered location. The plant's growth slows or stops. Leaves may die back, and to the inexperienced eye, it might look dead. But come spring, new growth emerges from the center, fresh traps unfurling, the plant renewed. This cycle of dormancy and growth connects the cultivated plant to its wild origins, a rhythm set by millennia of adaptation to seasonal climates. The physiological changes that occur during dormancy are complex. As temperatures drop and day length shortens, temperate carnivorous plants undergo metabolic changes that prepare them for winter. Carbohydrate reserves are moved from leaves into rhizomes or roots for storage. Growth points become protected by specialized scales or thick tissue. Cellular processes slow down, reducing the plant's energy needs to a minimum. These changes are triggered by environmental cues, particularly temperature and photoperiod. Plants can sense day length through photoreceptor proteins that detect the ratio of light to dark hours. They can sense temperature through changes in membrane fluidity and enzyme activity. When these cues indicate that winter is approaching, the plant initiates its dormancy program. Attempting to keep a temperate carnivorous plant actively growing through winter by providing warmth and long days disrupts this program and eventually harms the plant. Carnivorous plants can be propagated in various ways. Carnivorous plants reproduce both sexually, through seeds, and asexually, through vegetative means. Seeds offer genetic diversity, each seedling slightly different from its parents. But seeds can be slow to germinate and require specific conditions. Many carnivorous plant seeds need cold stratification, a period of cold and moisture to break dormancy. Some need light to germinate. Others require smoke or specific soil fungi. Vegetative propagation is often easier and faster. Many carnivorous plants produce offshoots or divisions that can be separated and potted individually. Venus flytraps form clumps over time, each rosette capable of becoming an independent plant. Sundews can be propagated from leaf cuttings. A single leaf laid on moist soil can sprout new plantlets along its edges. Some pitcher plants produce runners or rhizomes that spread underground, each segment capable of generating a new plant. Tissue culture, a laboratory technique, allows mass propagation of carnivorous plants from tiny pieces of tissue. This method is used commercially to produce large numbers of genetically identical plants. It has also been valuable in conservation, allowing rare species to be multiplied without removing more plants from the wild. Tissue culture requires sterile conditions and specialized nutrients, but it can produce hundreds or thousands of plants from a single parent. For hobbyists, propagation is a rewarding way to expand a collection or share plants with others. Watching a leaf cutting develop roots and tiny traps, or separating a cluster of Venus flytraps into multiple plants, connects the grower more deeply to the life cycle of these organisms. It transforms passive observation into active participation, a partnership between human and plant, each benefiting from the other's efforts. The success rates of different propagation methods vary by species. Venus flytraps propagate readily from leaf cuttings, with success rates often exceeding 50% under good conditions. Sundews are even easier. with some species producing plantlets from nearly every leaf cutting. Pitcher plants are more challenging. While they can be divided, leaf cuttings rarely succeed, and tissue culture is often the preferred method for mass propagation. Seed propagation offers the advantage of genetic diversity, which is important for long-term population health. However, many carnivorous plant seeds have special requirements that make them difficult to germinate. Cold stratification mimics winter conditions, breaking the seed's dormancy. Some seeds require this treatment for several months before they will germinate. Others need exposure to smoke chemicals, adaptations to fire-prone habitats, where germination after fire provides seedlings with a temporarily competition-free environment. Carnivorous plants have been used in traditional medicine. Throughout history, humans have used plants for medicine, and carnivorous plants are no exception. Various cultures have employed pitcher plants, sundews, and other species to treat ailments. The roots of North American pitcher plants were used by some indigenous tribes to treat smallpox and intixious, tuberculosis, made into infusions, or poultices. Sundews were used in European folk medicine as cough remedies, their sticky secretions thought to soothe respiratory inflammation. The effectiveness of these treatments is uncertain. Modern pharmacological analysis has found some compounds in carnivorous plants with potential medicinal properties, including antibacterial and anti-inflammatory substances. Some sundews produce compounds called plumbagin and naphthoquinones, which have shown antibacterial activity in laboratory studies. Whether traditional preparations contain sufficient quantities of these compounds to be medically effective remains unclear. What is clear is that humans have long recognized carnivorous plants as unusual and potentially powerful. Their ability to digest flesh, even insect flesh, may have suggested medicinal potency to traditional healers. The distinction between a plant that consumes animals and an animal that consumes plants blurs in carnivorous plants, perhaps making them seem imbued with qualities of both kingdoms, neither fully plant nor fully something else. Today, carnivorous plants are not widely used in medicine, and harvesting them from the wild for any purpose is discouraged due to conservation concerns. But the history of their use reminds us that humans have always sought to understand and utilize the natural world, looking to plants for cures, sustenance, and inspiration. Carnivorous plants, with their striking forms and unusual habits, have occupied a special place in this relationship, admired and feared, utilized and protected. The antibacterial properties of sundews have attracted modern research interest. The compound plumbagin, found in many sundew species, has been shown to inhibit the growth of various bacteria, including some antibiotic-resistant strains. This has led to investigation of sundews as potential sources of new antibiotics. However, the low concentrations of plumbagin in the plants and the difficulty of sustainable harvesting make it unlikely that sundews will become commercial sources of medicine. Other carnivorous plants have also yielded interesting compounds. Some pitcher plants produce enzymes that could have industrial applications in waste treatment or food processing. The digestive enzymes of carnivorous plants are optimized to function in the specific conditions of the trap, often acidic, sometimes cold, which might make them useful in industrial processes where conventional enzymes don't work well. Research in this area is ongoing, though still in early stages. Climate change threatens carnivorous plant habitats. As global temperatures rise and weather patterns shift, carnivorous plants face new challenges. Many species occupy narrow climatic niches, adapted to specific temperature ranges, precipitation patterns, and seasonal cycles. Changes to these conditions can be catastrophic. Warmer winters may prevent necessary dormancy periods. Altered rainfall can dry up bogs or, conversely, flood habitats beyond tolerable levels. Rising temperatures may allow competitor species to invade habitats previously too harsh for them. Coastal carnivorous plant populations face threats from sea level rise. Many bogs and wetlands exist in low-lying coastal areas. As oceans rise, saltwater intrusion kills freshwater plants, including carnivorous species. Some populations have already been lost, their habitats submerged or rendered uninhabitable by salt. Others cling to survival in increasingly marginal conditions. Fire patterns are changing as well. Some carnivorous plant habitats, particularly in the southeastern United States, are fire-adapted ecosystems. Regular, low-intensity fires clear competing vegetation, maintain open conditions, and trigger seed germination in some species. Fire suppression, practiced for decades to protect human property, has allowed these habitats to become overgrown, shading out carnivorous plants. Conversely, more intense fires, driven by climate change, can sterilize the soil, killing the plants outright. Adaptation to climate change is possible for some species through migration, shifting their ranges to follow suitable conditions. But carnivorous plants often cannot migrate easily. They require very specific soil conditions, water quality, and microclimates that are not easily found. Additionally, habitat fragmentation due to human development creates barriers. Isolated patches of suitable habitats surrounded by farms, roads, and cities that carnivorous plants cannot cross. The interaction between climate change and habitat fragmentation is particularly concerning. As climates shift, species need to move to track suitable conditions. But fragmented landscapes prevent movement, trapping populations in place even as conditions become unsuitable. For carnivorous plants, which often already occupy small, isolated habitats, this double threat is especially severe. Some carnivorous plant species have very narrow geographic ranges, sometimes occurring in only a handful of locations. These endemic species are particularly vulnerable to climate change. If their entire range shifts outside of suitable climate conditions, they may go extinct, with no possibility of migration to new habitats. Conservation efforts for these species may require active translocation, moving plants to new locations where suitable conditions are expected to persist under future climate scenarios. Research on carnivorous plants continues to yield surprises. Despite their long study, carnivorous plants continue to surprise researchers. Recent discoveries include the finding that some pitcher plants use rain to help trigger trap closure, the mechanical impact of raindrops on the pitcher rim knocking insects off balance. Others have revealed that certain sundews can distinguish between nutritious prey and non-nutritious debris, responding more strongly to items that contain nitrogen. Genetic sequencing has uncovered the evolutionary history of carnivorous plants in unprecedented detail, revealing which genes were co-opted for carnivory and how similar genetic changes occurred independently in different lineages. This research has implications beyond botany, informing our understanding of how complex traits evolve and how genomes respond to environmental pressures. Studies on the microbiomes of pitcher plants have revealed diverse bacterial communities that vary between species, locations, and even individual pitchers. These communities play crucial roles in digestion, breaking down prey more efficiently than the plant could alone. Understanding these relationships is shedding light on symbiosis, cooperation, and the invisible ecosystems within ecosystems. New species continue to be discovered. Remote regions of Southeast Asia, South America, and even Australia occasionally yield carnivorous plants previously unknown to science. Each discovery adds to our knowledge of the diversity and distribution of carnivory. revealing new variations on familiar themes. As long as wild places remain unexplored, and as long as scientists continue to look closely, carnivorous plants will continue to reveal their secrets. One recent surprising discovery involves the role of ultraviolet light in prey attraction. Some pitcher plants were found to have UV reflective patterns on their pitchers that are invisible to human eyes, but visible to insects. Many insects, including flies and bees, can see UV light, and they use UV patterns to locate flowers. Some pitcher plants apparently exploit this sensory system, using UV patterns to make their traps more attractive to insects searching for flowers. Another area of active research involves the chemistry of pitcher plant fluids. The composition of these fluids is more complex than previously realized, containing not just digestive enzymes, but also compounds that affect insect behavior, wetting agents that break down the water repellent coating on insect cuticles, and pH buffering systems that maintain optimal conditions for digestion. Some of these compounds appear to be produced by bacterial communities rather than by the plant itself, highlighting the importance of symbiotic relationships. The beauty of carnivorous plants lies in their duality. At their heart, carnivorous plants are beautiful contradictions. They are rooted yet predatory. They are plants yet hunters. They are gentle in appearance yet deadly in function. This duality is part of their enduring appeal. They challenge our categories, forcing us to think about what it means to be a plant, what it means to be a predator, and where the lines between different forms of life truly lie. In their stillness, they embody patience. A pitcher plant may wait weeks for a single insect to fall into its trap. A Venus flytrap remains open for days, its jaws set, ready to snap shut in an instant, but only if the right conditions are met. This patience is alien to us, accustomed as we are to motion and immediacy. Yet it is effective, proven by millions of years of survival. In their traps, they embody beauty and death entwined. The glittering dew of a sundew is alluring precisely because it is deadly. The deep throat of a pitcher plant, colorful and mysterious, draws the eye even as it draws the fly. There is an aesthetic completeness to these plants, form following function so perfectly that the result is art. Nature, in crafting them, created sculptures that happen also to be highly effective predators. Perhaps this is why we are drawn to carnivorous plants. They are windows into a way of being utterly different from our own, yet understandable. They solve problems, adapt, survive. They are beautiful and terrible, familiar and alien. In growing them, observing them, learning about them, we touch something fundamental about life itself, its creativity, its persistence, and its endless capacity to surprise us. The aesthetic appeal of carnivorous plants has made them popular in cultivation, but this appreciation can be a double-edged sword. On one hand, widespread cultivation reduces pressure on wild populations by providing legal sources of plants for enthusiasts. It also raises awareness of these species and their conservation needs. On the other hand, the popularity of carnivorous plants has driven illegal collection and trade, threatening wild populations. The contradiction of carnivorous plants, beautiful yet deadly, plants yet predators, has philosophical implications as well. They challenge the clean distinctions we like to draw between categories of life. Are they more plant-like or more animal-like? The question itself may be misguided, reflecting our need to categorize rather than any inherent property of the organisms. Carnivorous plants simply are what they are. Organisms that have evolved specific adaptations to specific challenges, fitting into no category perfectly, but embodying something of many. Carnivorous plants remind us of nature's ingenuity, As we begin to draw our gentle journey through the world of carnivorous plants toward its close, we are left with a profound sense of wonder at nature's ingenuity. In the face of scarcity, where most plants would struggle and fail, carnivorous plants found a different path. They looked beyond the soil, beyond the conventional sources of nutrition, and discovered that the air itself was full of food, if only it could be captured. The mechanisms they evolved, the traps, the enzymes, the lures, are elegant solutions to complex problems. They demonstrate that evolution is not random chaos, but a process that produces sophisticated, functional designs. Each carnivorous plant is a testament to the power of natural selection, millions of small changes accumulating over vast timescales into something remarkable and new. These plants also remind us of the importance of diversity. Carnivorous plants occupy a small niche in the vast spectrum of plant life, but their existence enriches the world. They provide habitat, alter nutrient cycles, inspire scientific inquiry, and captivate human imagination. A world without them would be measurably poorer, not just ecologically, but aesthetically and intellectually. As you lie here, letting your mind wander through bogs where sundews sparkle and pitcher plants stand like sentinels, imagine the quiet drama unfolding there. A fly lands on a sundew's glittering leaf and finds itself held fast. Tentacles slowly bend inward, embracing their captive. In a nearby pitcher, water ripples as something struggles within. A Venus flytrap sits open, waiting, counting touches, ready to snap shut in an instant. And all around, in the soil, in the water, in the air, life continues its ancient dance, each organism playing its part in the endless story of survival. Let these images soothe you. The patience of the pitcher plant, waiting for rain and prey, the delicate beauty of the sundew, jeweled with false dew, the sudden violence of the Venus flytrap, a snap and then stillness again. The tiny Bladderworts, invisible but present, their traps firing in fractions of seconds in the microscopic world beneath the water's surface. All of this continues, whether we watch or not, whether we understand or not. The carnivorous plants grow, trap, digest, and reproduce, year after year, season after season, in quiet corners of the world where few humans venture. They are ancient and modern, specialized and successful, beautiful and deadly. As your breathing slows and deepens, as your thoughts drift and dissolve, imagine yourself shrinking down, becoming small enough to walk among these botanical hunters. The leaves tower above you, like trees. The pitcher plants are vast wells, their openings like cavern mouths. The sundew's tentacles are forests of crystalline columns, each droplet a sphere larger than your head. At this scale, the carnivorous plant world becomes fantastical, a realm where the rules are different and the boundaries between plant and predator blur into something new. But you are safe here, in this imagined space. You can observe without danger, marveling at the machinery of capture and digestion, the chemistry of enzymes and acids, the physics of trap closure and tentacle movement. You are a visitor to a world that operates on principles foreign to your daily experience, yet somehow comprehensible, somehow beautiful. And now, as the journey ends and sleep beckons, let the carnivorous plants fade gently from your mind. Let them become dream images, soft and indistinct, their shapes merging with the darkness behind your eyelids. The sundew's tentacles become stars in a night sky. The pitcher plants become wells of shadow, inviting and mysterious. The Venus flytrap's jaws become smiles, opening and closing slowly, rhythmically, like breath itself. You are drifting now, floating between waking and sleeping, between thought and dream. The Carnivorous plants watch over you as they watch over their own domains, patient and still, waiting for whatever comes next. In their presence, there is no urgency, no demand, only waiting, only patience, only the slow unfolding of time. Let go now. Let the images dissolve. Let your body sink deeper into comfort, your mind drift further into peace. The carnivorous plants will still be there tomorrow, in bogs and wetlands, on windowsills and in terrariums, continuing their ancient work. But for now, they release you into sleep, into dreams, into the vast and gentle darkness, where all thoughts eventually come to rest. Breathe slowly. Feel the weight of your body. Notice how still you have become, how quiet. You are like a carnivorous plant yourself now, rooted, peaceful, simply existing. No effort is required. No action is needed. You are held by stillness, embraced by calm, sinking deeper and deeper into rest. And in the morning, when you wake, the world will be there again, full of wonders large and small, of plants that trap and plants that grow, of mysteries solved and mysteries remaining. But for now, sleep, just sleep. The carnivorous plants send you their blessing, these hunters of the botanical world, these patient waiters, these beautiful contradictions. They ask nothing of you but that you rest, that you dream, that you let go of waking thoughts and surrender to the gentle pull of sleep. Close your eyes if they are not already closed. Feel yourself sinking, drifting, dissolving into comfort and darkness. The journey is complete. The story is told. And now, only sleep remains, soft and welcoming, pulling you down into its embrace. Good night. Sleep well. And may your dreams be as gentle and patient as a sundew waiting for dawn, as quiet as a pitcher plant in the rain, as peaceful as a bog at twilight, where carnivorous plants stand like guardians of stillness, watching over the slow passage of time.